Okay, let's wait another 30 seconds and then we slowly start. Okay, I think we can, sorry, stop share. Voila. Oops. Voila. So let's start. We waited a minute. So good afternoon, everyone. Welcome to this edition of the NITheP Colloquium. My name is Francesco Petruccione, and today I have a double pleasure in introducing you today's speakers. Because not only because she will speak about a very interesting topic, also because she's working on her PhD in our research group at UKZN. So today's speaker is Betony Adams, and as I mentioned, she's completing her PhD. She did her master's also at UKZN, working on applying methods of the theory of open quantum systems to quantum effects in biological system, and in particular in avian migration on the avian migration problem. Yeah, but today she will speak about something slightly different, which we became interested in during her PhD, which is quantum effects in the brain. And a few months ago, probably maybe half a year ago, I don't remember exactly, Betony published a very nice review article on quantum effects in the brain that was very well received and is already cited a lot. And for that reason, we ask her today to give us a brief summary of that very extensive review article. Yeah, and like in most NITheP webinars, Dr. Sinaisky kindly offers to be the co-host and moderate the questions at the end of the presentation of Betony. So please, if you have questions, make use of the Q&A facility at the bottom right of your Zoom window, and Ilya will moderate and coordinate the questions. So Betony, if you would like to start sharing your screen, we are all very curious to listen to your presentation. So thank you very much, Betony, for agreeing to be with us today.
Thank you so much, Francesco. Let me just see. Can you see my screen?
Yes, perfectly. Thank you very much.
Okay, great. Thank you so much for that introduction and for the opportunity to speak. As already mentioned, I'm Betony Adams, and I'm doing my PhD in the Center for Quantum Technology at UKZN under the supervision of Prof. Francesco Petruccione and Dr. Ilya Sinaisky. Our group is probably better known for quantum machine learning, but we also do some quantum biology, which I'll be talking about today, particularly about a subject which is sometimes seen as controversial. That is whether quantum effects may play any role in how the brain works. I've heard it said fairly often that just because quantum physics and consciousness are both difficult to understand doesn't mean that they are necessarily related. I'm not going to be focusing all that much on trying to explain consciousness. Rather, I'm going to present evidence for whether quantum effects might be implicated in the physiology of the brain or the nerves that make up the central nervous system. I won't be going into the specifics of my own research, but will rather give a broader introduction to the subject and an overview of how confident we can be in suggesting that the brain utilizes quantum effects. To give a basic framework for the presentation, I'll start with a brief introduction to quantum physics and the specific effects I will be talking about. I'll then introduce what I mean by quantum biology and quantum neurobiology and how the two are related before moving on to the specific examples of general anesthetics, neurotransmitters, and neural entanglement. And I'll end off with why I find this field of research really interesting and potentially important. For many of you, this slide will probably all seem very familiar and possibly simplistic. My aim is only to give a general idea of the development of quantum ideas without getting lost in the details. Could be said that quantum mechanics developed as a way to explain experimental results and physical phenomena that couldn't be explained using classical theory. For instance, in order to explain the spectral energy distribution of black body radiation, which was known as ultraviolet catastrophe, Max Planck in 1900 introduced the idea that interactions between matter and electromagnetic radiation are quantized, with H, or Planck's constant, used as a measure of this quantization. And many of you will recognize this idea of quantization as the origin of the term quantum. 5 years later, Einstein built on this idea of quantization by introducing the idea of quantized light, which we know as photons, an idea he used to explain the photoelectric effect. Niels Bohr's 1913 model of the hydrogen atom, which replaced classical electron orbits with quantized energy levels, and Compton's work on X-ray spectroscopy, all contributed to the emerging quantum era in modern physics. While Planck and Einstein began the revolution by suggesting that radiation, which was conventionally understood to be a wave, sometimes behaves like a particle, de Broglie made the complementary suggestion that matter might behave like a wave. This was later demonstrated by the interference patterns of electrons. Quantum mechanics, which began as a way to explain experimental results that did not make sense classically, was later formalized by physicists such as Schrödinger and Heisenberg, who gave us the maths that we use to describe and predict the dynamics of microscopic quantum systems with such accuracy today. So during the course of this talk, I will probably make use of the terms quantum system, quantum state. I might also talk about quantum models or ways of modeling the state of a system. I thought it would be useful to clarify just what I mean by these terms. When I talk about a quantum system, I'm basically talking about the thing or the object that I'm interested in. In quantum physics, the focus is microscopic, so my system might consist of electrons or photons or atoms and the interactions between these. A quantum state then describes the physical properties of the system. Very simplistically, just as a classical object, such as a car, has an energy or momentum that can be measured, your quantum system, your electron or photon or atom, has similar properties that are described using their state vector. What is interesting about quantum mechanics is that for two quantum states describing a system, say for example spin up and spin down, where spin describes the action of a particle in a magnetic field, a linear combination of these states also describes the system. Take for example the state given on the slide. Psi, the state vector, could describe an electron with some probability of being in the spin up state and some probability of being in the spin down state. This is known as the superposition principle, the inspiration for Schrödinger's cat's thought experiment. Finally, what do I mean when I talk about quantum models? This is just a way of describing, using maths, what happens to a state over time. How does it change? If you were looking at a classical system like a car, you might use Newton's equations to describe how the car's state changes. In quantum mechanics, you would probably use Schrödinger's equation or other related models developed over the history of quantum mechanics to describe how the state of, say, an electron changes. Now that I've hopefully clarified what I mean by quantum system, state, and model, I want to be clear about what exactly these quantum effects I keep talking about are. I've already mentioned Bohr's model of the atom and how atomic structure is described by quantum mechanical principles. In this sense, all biology might be described as quantum because biological systems are made up of molecules and atoms. This is what is known as trivial quantum effects in quantum biology because they don't really let us say anything new or interesting about biological systems. What we are more interested in is what people describe as non-trivial quantum effects. I'm not going to go into any great detail as to what these are, but wanted to introduce them so you recognize them when they come up in the biological context. The first of the three main quantum effects I'll be discussing in this presentation is quantum coherence. I've already talked about the fact that quantum systems can be in a superposition of different states. Well, quantum coherence describes how the states in a superposition are related. Quantum tunneling, which is another of the non-trivial effects I will be discussing, is the non-zero probability for a quantum particle to pass through a classically forbidden energy barrier. And finally, entanglement, which is the non-classical correlation between two quantum states. If any of you have done any reading on quantum biology or quantum computing, you will have no doubt heard about this thing called decoherence, which refers to the loss of quantum effects in a system. For this reason, quantum systems have conventionally been described as isolated and at low temperatures because interaction with an environment speeds up decoherence. But biological systems, perhaps by definition, have to interact with the environments, and they exist at physiological temperatures. This means that we can't apply models that are developed for conventional quantum systems to biological examples. But physicists have already developed a way of modeling how a quantum system might interact with an environment. This is known as open quantum systems. This open systems approach is used by many researchers to describe quantum effects in the biological context. To simplify, the subject under study is broken down into the system of interest, which might be electrons moving through a leaf, the environment these electrons interact with, which might be modeled as oscillating particles, and then the interaction between the two. So what then exactly is quantum biology? The question of whether physics might contribute to understanding biology is not a new one. And the question of whether quantum physics might have anything to offer biological studies has a history almost as old as quantum physics itself. Schrodinger's famous 1944 lecture series, What is Life? The Physical Aspect of the Living Cell, is often used as a milestone in quantum biology. In it, he describes an aperiodic crystal responsible for hereditary traits that later inspired studies of DNA. Before this, however, Bohr had already given a lecture called Light and Life, and other physicists such as Pascal Jordan had already coined the phrase quantum biology. Of the non-trivial quantum effects I will discuss in this presentation, quantum tunneling was the first to make an appearance in the 60s already in the context of enzymes. A decade later, coherent spin dynamics in radical pairs was proposed as a way to describe magnetoreception. And last, but experimentally most successful of the topics recognized as quantum biology is coherent energy transfer in photosynthesis. Pretty much the main objection raised against the possibility that quantum effects might play a role in biological systems is this phenomenon of decoherence. As I've mentioned before, quantum effects are delicate and easily destroyed by interaction with an environment, whereas biological systems unavoidably interact with their messy environments. However, the field of quantum biology, as we understand it today, has made some progress towards establishing that quantum effects might play a role in biological systems. And it has arguably had most of its successes in the context of three different biological phenomena: photosynthesis, olfaction, and magnetoreception. In the case of photosynthesis, energy and electron transfer in plants has been suggested to employ quantum coherence. This, despite some contention, has been supported by experimental findings. Quantum tunneling has a long history in quantum biology, having first been observed in enzymes. Its most famous application is arguably to olfaction, or the sense of smell. And finally, the spin dynamics of radical pairs in birds' eyes have been used to explain some of the behavioral aspects of the avian compass. It's on this foundation that quantum neurobiology has been based. So finally, we get to the neurobiology. This simplified diagram of a nerve cell is a useful way to locate the quantum effects that I will be talking about in a neural context. So the parts of the nerve cell I will be referring to are as follows: the microtubules, which make up the cytoskeleton of all cells and are important in cell structure, transport, and cell division. The mitochondria, which are the site of electron transport chains, similar to those found in photosynthetic networks, and which are the primary site for the production of biophotons. Synapses, which allow nerves to communicate amongst each other and are the proposed site of endocytosis. And finally, briefly, the myelinated axon has been suggested to be a possible waveguide for biophoton transport. I've given you some idea of the history of quantum biology, but what about the history of quantum neurobiology? While quantum theory might have inspired various discussions about the nature of consciousness, the first detailed theory and outlining how quantum theory might be applied in the context of neurobiology emerged in the 90s. Roger Penrose, a physicist famous for work done in general relativity and cosmology, for which he won this year's Nobel Prize, published a book called The Emperor's New Mind, in which he suggests that classical rules cannot fully describe consciousness. What he lacked, however, was a biological site to locate any hypothetical quantum effects. Stuart Hameroff, an anesthesiologist, had been interested in microtubules for some time, and he suggested to Penrose that they might possibly support quantum effects. Together, they proposed the theory of orchestrated objective reduction, where quantum computations in microtubules in neurons alter axon firing rates and thus consciousness. I won't go into the full details of the theory. If you're interested, there's a very long, detailed 2014 review of the subject called Consciousness in the Universe. My focus in this talk is less about consciousness and more about the underlying physiological mechanisms that allow neurons to function. What I would like to focus on from the theory of orchestrated objective reduction is the biological site that Hameroff and Penrose chose to situate their theory in, which is microtubules. Microtubules are formed through the polymerization of tubulin dimers. Tubulin dimers consist of an alpha and a beta tubulin protein. Tubulin dimers form filaments, and then 13 of these filaments form a microtubule. What was interesting to Hameroff and Penrose about tubulin dimers is that they are potentially a way to realize a biological qubit. For those of you familiar with computer science, you'll know that a binary bit can have a value of 0 or 1. A qubit, on the other hand, is a quantum bit, and as I've already discussed, quantum states can exist in superpositions, in this case of 0 and 1. When orchestrated objective reduction was first developed, Hameroff and Penrose suggested tubulin dimers could exist in a superposition of conformational states. But the theory has since been updated to include electric and magnetic dipole states. That's all I'm going to say about orchestrated objective reduction, but I'll get back to microtubules in a bit. I like this quote because it sums up, at least for me, one of the methods by which it is possible to address the unsolved problem of consciousness, or on a lesser level, the physiology of the brain. And that is to study those chemicals or interventions that have an effect on consciousness or brain function. This approach will inform to a large extent this presentation as I discuss research relating to general anesthetics, neurotransmitters, and antidepressants such as lithium. This image is also borrowed from a talk given by Luca Turin. What it highlights is the extreme diversity of molecules that have anesthetic effects. As you can see, they range from the extremely simple xenon to the large and unwieldy looking olfaxolone. What is remarkable is their lack of a uniting feature, which is what led Luca Turin to ask whether there was some other way to find a link between these very different molecules. The same question had already been, to some extent, asked by Stuart Hameroff, who, if you remember, is an anesthesiologist himself. He hypothesized that anesthetics might work through electronic rather than chemical interactions. So what do I mean by electronic? Well, very simply, in this case, I mean having something to do with electrons. And a large part of the success of quantum biology has been in describing how energy or electrons move through a biological medium, such as a leaf, during photosynthesis. In particular, it has suggested that this movement uses quantum coherence. To simplify things, at least as how I understand it, what this means is that when energy is transferred between pigments known as chromophores, which react to light, The energy is shared or delocalized over sets of chromophores. So in the diagram, the chromophores are the molecules shown in the black circle. Each colored semicircle represents those chromophores that share energy, whereas between colors, energy is transferred classically. So if you take anything away from the slide to apply to the context of neurobiology, let it be that the arrangements of pigments, known as chromophores, these light sensitive pigments, in a protein possibly allow for coherent energy or charge transfer. What I'm trying to illustrate with this slide is how quantum neurobiology has borrowed from work done in quantum biology. Once again, we are interested in how energy or charge moves through a protein. But in this case, our biological context is microtubules in nerve cells rather than antenna complexes in plants. So tubulin proteins, which make up microtubules, have similar arrangements of chromophores to photosynthetic networks. This has led researchers in quantum neurobiology to suggest that not only do microtubules allow for coherent energy and charge transfer in nerve cells, but that anesthetics might bind to these proteins and disrupt this coherent transfer. And that's their mechanism of, that's the mechanism by which they work. Is there any experimental evidence for anesthetics being electronic rather than chemical agents? Lucatura, whose quote began this section, was also interested in the movement of electrons in proteins. To test his ideas, he focused on xenon, which is the simplest general anesthetic, and as he argued, has no shape or chemistry, but does have physics. For inspiration, he took the 1989 experiment by IBM, where a scanning tunneling microscope was used to write out IBM in single atoms. The xenon atoms in this case extended the conducting surface of the metal under the microscope. In a similar way, Turin argued, xenon bound to proteins could extend the highest occupied molecular orbits and in this way facilitate electric currents. So in order to test this, he looked for an increase in free electrons in fruit flies under the influence of anesthetic. The free electron signal was measured as an increase in spin using an electron spin resonance experiment. Spin, if you recall, is the attribute that particles like electrons have that makes them behave like magnets in a magnetic field. So applying a magnetic field causes a split in the possible spin energy states. Now, if a radio frequency source matches the energy of that split, then the radio frequency radiation will be absorbed, with the amount of absorption giving a measure of the number of spins. What they found was that anesthetics did cause an increase in spin, possibly corroborating the hypothesis that general anesthetics are implicated in electron currents in proteins. However, there's still quite a lot of contention as to the origin of the spin signal, not to mention the fact that these flies, as well as being under the effects of anesthetic, were frozen and starved of oxygen at the same time. I've mentioned quantum tunneling a number of times now in the context of enzymes as well as olfaction. To give you a better idea of what I mean by this, I thought I'd discuss the concept in the context of quantum biology before moving on to the neurobiological context. The lock and key or docking mechanism has been used to describe the working of a number of biological systems, enzymes and olfaction included. The basic idea is that the shape of a molecule, such as an odorant, matches the shape of a specific receptor, and the correct fit activates the receptor. But it has also been suggested that odor receptors, which are known as G-protein coupled receptors, might be activated by the vibrational modes of specific odor molecules using vibration-assisted quantum tunneling. So very simplistically, in order for a receptor to be activated, an electron needs to move from one level to the next. And if the vibrational energy of the odorant molecule matches the energy of this transition, then the electron moves levels and the receptor is activated. So if you remember nothing else from this slide, then hang on to the fact that lock and key mechanisms in biological systems might alternatively work through vibration-assisted quantum tunneling, which we will now look at in the neurobiological context. So as I've already mentioned, olfactory receptors are GPCRs, so these G protein coupled receptors. GPC receptors proliferate in the body, and I think something like 1/3 of all pharmaceuticals target these receptors. And the receptors that neurotransmitters bind to on nerve cells are also GPCRs. Neurotransmitters are chemicals that allow nerves to communicate at the synapses. The binding of neurotransmitters to their appropriate receptors controls the movement of charged ions across nerve cells membranes, causing changes in potential and the firing or not firing of neurons. So following from work done in olfaction, there's been some suggestion that neurotransmitters such as serotonin and histamine and adenosine bind to and activate receptors through vibrational assisted quantum tunneling rather than by virtue of their shape. The work, however, is still theoretical. For example, agonists that activate the same receptors as the neurotransmitter serotonin, such as LSD, share similar vibrational spectra, which is calculated using density functional theory. So while the research is largely theoretical, there has been some attempts to test the theory experimentally. The method of testing follows the example of olfaction, which employs selective deuteration. Selective deuteration involves replacing hydrogen with its heavier isotope deuterium, which has an extra neutron. There's some evidence that species as diverse as fruit flies, cockroaches, and whitefish can smell the difference between hydrogen and deuterium. And this could potentially support the case for vibration-assisted quantum tunneling because deuteration changes the vibrational spectrum of a molecule, but not its shape. So how have neurotransmitters fared in similar experiments? Well, serotonin, which theory suggested is a good fit for vibration-assisted tunneling, with agonists of the serotonin receptors sharing vibrational spectra, unfortunately failed experimental tests, but this is, well, I should say experimental test thus far. Selective deuteration did, however, make a difference to the action of histamine, but it is not clear whether this is due to vibration-assisted tunneling or some other phenomenon. More experiments, no doubt, need to be done, as selective deuteration is a delicate process, and vibrational spectra can be altered by experimental mistakes, such as the unwitting inclusion of contaminants. So far, I've discussed 2 out of 3 of the main phenomena associated with quantum biology and how progress made in understanding these biological processes has been applied to understanding related neurobiological phenomena. In this third case, we are interested less in the movement of electrons than in their spin dynamics and how spin might translate into chemical or biological signaling. To recap, spin is the property that particles such as electrons or nuclei have that makes them behave like magnets in a magnetic field. So it's been known for quite some time that spin dynamics can have an effect on chemical reactions, and this was adapted to explain how birds might be able to sense the Earth's magnetic field. This mechanism, which is known as the radical pair mechanism, can be summarized in the following 3 steps. So first, a photon transfers its energy to an electron and an electron pair, causing that electron to be donated from a donor to an acceptor. So now you've got a spatially separated but spin correlated electron pair. And what I mean by spin correlated is that the spins of each electron are arranged in some specific way with respect to each other. In this case, the spins start off in what is known as a singlet state. Then, under the influence of the Earth's magnetic field and the surrounding nuclei, these spins switch between singlet and triplet states. If they happen to recombine during this time, the recombination product depends on whether they are in a singlet or triplet state. So how does the radical pair mechanism offer any insights into quantum neurobiology? Well, one of the recent hypotheses put forward in the field also depends on a spin-mediated physiological outcome. In this case, unlike with the radical pair mechanism, we are interested in nuclear spin, not electron spin, but the idea is similar. Physicist Matthew Fisher was interested in whether quantum physics could play a role in correlated neural firing patterns. So he hypothesized a way in which the entangled nuclear spins of phosphorus nuclei could result in entangled neural activation using molecules known as Posner clusters, or otherwise known as amorphous calcium phosphate. I'll be discussing this in further detail in the next few slides, but what I wanted to highlight here is that once again, we are looking at how spin correlation might result in a biologically relevant outcome. So how did Fisher get interested in this subject? Well, as the story goes, he had a period of depression, which got him interested in antidepressants and mood stabilizers, such as lithium, which is used to treat bipolar disorder. And similarly to the argument that Luca Turin made about xenon, lithium is one of the structurally simplest antidepressants. And he hypothesized that if neural processes used quantum effects, then drugs that target neural processes might have different neurophysiological effects for different isotopes. If you recall, isotopes are variations of elements with different numbers of nuclei. When Fisher looked for evidence of this, he found that experiments had been done using lithium isotopes, lithium-6 and lithium-7, which showed that lithium-6 promoted maternal behavior far more strongly than lithium-7 or the control group in the mothering behavior of rats. Even more recently, a similar effect has been shown for xenon isotopes. If you recall, xenon is the simplest of the general anesthetics. It has 9 stable isotopes. 7 of these have 0 nuclear spin, but there are 2 that have non-zero spin. Xenon 129 has spin half, and xenon 131 has spin 3/2. So researchers found that they used writing reflex as a measure of consciousness, and they found that xenon with non-zero nuclear spin was a much less potent general anesthetic than those with 0 nuclear spin. I think this is a pretty incredible result. Something as tiny as a neutron can have such a profound effect as to alter states of consciousness. And if you recall our previous discussion on isotopes in the context of vibration-assisted tunneling, some of you might be thinking that this isotope-dependent effect has something to do with changing the mass and thus the vibration by adding neutrons. But the researchers checked, and the effect seems to be independent of mass changes, which means it's most likely to have something to do with spin. Because changing the number of neutrons also changes the spin of the nucleus. So this interest in spin led Matthew Fisher to hypothesize that spin-mediated reactions might play a role in neurons. At first, he had to decide on which element would be best suited to act as a neural qubit. I've talked about decoherence before and how it causes loss of quantum effects. So in order to have any effect on neural firing, or more ambitiously, memory storage and retrieval, Fisher needed a qubit where decoherence happened very slowly. To give you some sense of timescales, I've included some timescales for quantum effects in biological contexts. As you can see, these range from picoseconds to hours. So how did Fisher propose to achieve quantum effects lasting for hours? To do this, he settled on phosphorus nuclear spin as the physical site of his proposed quantum effects. Phosphorus has a nuclear spin of a half. So for spin half nuclei, decoherence happens much more slowly because there's no quadrupole interaction with surrounding electrons. There are not so many biochemical elements with nuclear spin of a half, just hydrogen and phosphorus. Fisher calculated that quantum effects using phosphorus nuclei could last as long as 21 days. However, there's been some contention with other researchers who calculated that this would most likely be closer to 37 minutes, which is still a long time for coherence to last. So what exactly is an entangled plasma molecule? How do you create one, and how does this spin entanglement translate to neural firing? To start, you need ATP. ATP is the molecule from which cells derive their energy and is the end product of these electron transport chains that I keep talking about. It is created in mitochondria and is present in all cells in the body. From ATP, you get pyrophosphate, which is 2 phosphate ions. Phosphate ions are made up of phosphorus surrounded by oxygens. So the next step is the creation of phosphates from pyrophosphates. And this is the important step for creating the entanglement. Fisher argues that if this phosphate production happens through the influence of an enzyme called pyrophosphatase, then the molecular dynamics mean that the phosphorus nuclei end up in a singlet entangled state. If you recall from previous slides, a singlet is a specific orientation of spins with respect to each other. If these phosphorus spins now interact with other spins in the environment, then this will cause decoherence. So in order to protect the phosphorus spins, they need to be in an environment with as little interaction as possible. So Fisher suggests that this is possible if the phosphates bind with calcium to form this amorphous calcium phosphate, otherwise known as plasma clusters. The oxygens in the phosphates and the calciums in the plasma clusters all have 0 nuclear spin, which means they form like a protective cage around the phosphorus nuclei and allow for these long coherence times predicted by Fisher. So then how do these entangled phosphate nuclear spins, phosphorus nuclear spins, have any effect on neural action? Fisher imagines that if entangled phosphates end up in different plasma molecules and then get taken up into different neurons during synaptic endocytosis. Under a change of pH, the plasma molecules bind, and when they bind, they melt and they release calcium ions. And calcium ions are integral to neurotransmitter release. So Fisher contends that entanglement can affect the binding of plasma molecules, which in turn will affect the release of calcium and the downstream action of neurotransmitters. While this still has to be tested experimentally, it also offers a way of imagining how lithium isotopes might have an effect on neural action. It's been suggested that if you replace the central calcium in a plasma molecule with a lithium ion, then the different spin dynamics may have an effect on the entanglement and thus the binding rates of plasma molecules and subsequent calcium production and neural activation. What is also potentially interesting about the proposed plasma molecule mechanism is its similarities to ideas put forward in quantum computing. So one of the challenges of quantum computing is the physical realization of qubits. Various qubit alternatives have been explored, from superconducting circuits to trapped ions. One of the first suggestions for an implementable, scalable quantum computer was made by Bruce Kane in 1998. He proposed that information could be encoded onto the spin-half nuclei of phosphorus and a substrate of spin-0 silicon, similarly to Matthew Fisher's phosphorus nuclei in their spin-0 cage of oxygen and calcium. Spin-based quantum computing is still one of the main avenues being pursued in the quest for scalable quantum computers, particularly in Australia. So I've been through some of the details about how quantum effects might play a role in effective neural processing, but to end off the presentation, I wanted to talk about why I find this field so interesting and potentially useful. So I was reading an article was last year about the effects that serotonin is having on a species of shrimp. Serotonin is a neurotransmitter. I think, in fact, apart from dopamine, I think it's probably the most famous of the neurotransmitters, being partly responsible for what we call happiness. So many antidepressants are SSRIs, which stands for selective serotonin reuptake inhibitors. And millions of people take these drugs, which means that our water supplies are beginning to have measurable concentrations of SSRIs in them. Recently, it was discovered that exposure to SSRIs was causing the death of a certain species of shrimp. This was because the shrimp, which ordinarily hid in darker waters, was swimming towards the light and getting eaten in larger numbers by the fish. And why I find this interesting is that the molecule that makes up serotonin is tryptophan. Tryptophan is one of those light harvesting pigments called chromophores that I've been talking about in the context of photosynthesis and general anesthetics. Tubulin, for example, the protein that makes up microtubules, has 8 tryptophan chromophores embedded in it. I think it's really amazing that our consciousness, as mediated by neurotransmitters such as serotonin, might be in some sense responsive to light. In 1932, Niels Bohr gave a lecture called Light and Life for the opening of the International Congress of Light Therapy in Copenhagen. Light and electromagnetic radiation was central to the development of quantum mechanics. Out of Einstein's many groundbreaking ideas, it was photons and the photoelectric effect that won him the Nobel Prize. Light is also central to life, with photosynthesis being responsible for a large percentage of Earth's biomass. So it's no surprise that progress made in understanding light might have been interesting in the context of biology. But while it's easy enough to find Bohr's lecture where he discusses what the new discoveries of quantum theory might mean for the study of living organisms, it's more difficult to trace what happened to the therapy part of the Congress. The meeting was devoted to the role of light in biology, biophysics, and therapy. The role of light in biophysics has subsequently incorporated quantum theory and the study of photosynthesis, but the idea of light therapy seems to have fallen away after that initial Congress. We know for sure that that plants interact with light, but it is less certain if or how humans might interact with light. And this, to some extent, has now re-emerged as a growing field of research. So quantum mechanics deals in great detail with light-matter interactions. But what can it tell us about light and living matter interactions? The diagram shown here illustrates the electron transport chains of photosynthesis and respiration. Electron transport chains are integral to living organisms as they create the electrochemical gradient that is fundamental to creating ATP. And ATP is the molecule that cells derive their energy from. As you can see in the bottom section of the diagram, which is relating to photosynthesis, light is harvested by certain complexes in the electron transport chain, and this energy is transferred through the chain to result in ATP synthesis. The top diagram is a similar electron transport chain that is found in mitochondria and present in all of our cells. These similarities are potentially interesting in the context of transcranial photobiomodulation, which is the application of red or near-infrared light to the cranial area. And the proposed target of this light is one of the complexes that make up the mitochondrial electron transport chain. Is it possible? Then in some sense, we also have the biological apparatus for photosynthetic mechanisms. Can quantum mechanics, which has been successfully applied to photosynthesis, help explain light and matter interactions in a similar context in mitochondria? Light therapies such as photobiomodulation have shown success in treating a variety of brain disorders, ranging from depressions and schizophrenia to brain damage. It has also been shown to improve memory, learning, and executive function. And South Africa is not so far behind this trend either. I recently saw that Carte Blanche had done a show on a Durban neurologist who has successfully treated patients with Parkinson's and brain damage using photobiomodulation. Quantum dots, which are nanoscale semiconductors with quantum properties that can emit light, have also been shown to be a promising therapy for undoing the protein clumping that causes Alzheimer's and Parkinson's. So what I find possibly even more interesting is that not only might we harvest photons, but our cells actually produce light, biophotons, which are ultra-weak, near ultraviolet to near infrared photons. The site of biophoton production seems to be the mitochondria, and though it is still a new area of research, there's some suggestion that biophotons may be an alternative form of neural signaling, with researchers hypothesizing that the myelinated axon could act as a photon waveguide. Do biophotons offer us any insight into the brain? Well, Sylvia Plath wrote during one of her periods of mental illness, this is the light of the mind, cold and planetary. The trees of the mind are black, the light is blue. But recent research suggests that the light of the mind is not blue, but possibly red. A recent experiment found that when slices of brain from species ranging from bullfrog, mouse, chicken, pig, monkey, and human were stimulated with glutamate, the frequency of the biophotons they emitted ranged according to the intelligence of the species. While the measure of intelligence is still debatable and correlation does not mean causation, it appears as though higher intelligence corresponds to redshifted lower frequency biophotons. Biophotons are also related to reactive oxygen species, with mitochondrial reactive oxygen species hypothesized to be the source of metabolic biophotons. Reactive oxygen species are highly reactive and can cause damage to cells. They are implicated in a number of cellular malfunctions, such as DNA damage, disease, mental illness, and aging. When people talk about the importance of antioxidants, their role is in countering the effects of these reactive oxygen species. However, reactive oxygen species are also essential in cellular signaling, playing a role in cellular regeneration amongst other things. Which brings us to another way in which quantum mechanics might contribute to our understanding of the brain. I've spoken about the quantum property of spin a number of times already, but it also appears that spin dynamics might play a role in reactive oxygen production and control. While it has been accepted that many animals have a magnetic sense, it was unclear whether humans have any way of sensing weak magnetic fields, such as the Earth's magnetic field. But recent experiments showed that changing geomagnetic fields disrupts alpha waves, which are the 8 to 13 hertz oscillations in brains. It has also been shown separately that biophoton increase is linked to alpha rhythm fluctuation. As biophotons are linked to reactive oxygen species, is it possible that reactive oxygen species play some role in this magnetic sensing? If you recall the discussion of the radical pair mechanisms in the bird's compass, I talked about the switching of these spin states between singlet and triplet states. So it's been shown with respect to reactive oxygen species that this singlet-triplet mixing can influence the production of reactive oxygen species. It has also been shown that magnetic fields at Earth strength can alter the spin mixing and thus alter reactive oxygen species. What is potentially interesting about this is that there's some evidence that geomagnetic storms, which cause small fluctuations in the Earth's magnetic field, have been linked to psychological effects such as increased suicides. Could this be an effect of disrupted reactive oxygen yields? On the other hand, reactive oxygen species are important signaling molecules involved in tissue regeneration processes. And studies have shown that magnetic fields can promote or restrict the regeneration of planaria, which is small flat bones that have been cut in half. So quantum mechanics is often adopted as a quasi-mystical theory open to all sorts of misinterpretations. What I thought was interesting, however, is that there is some evidence, though probably not enough to base any conclusions on, that things like meditation decrease biophoton emission. Could this point to a decrease in the reactive oxygen species and thus explain the beneficial effects of meditation? That's as much as I'm going to say about how quantum effects might play a role in the physiology of the brain or how nerves function, but I thought I'd end off with a very brief discussion of how we might extend this to address human behavior. Niels Bohr, one of the founders of quantum theory, also felt strongly that the formulation of quantum theory might have something to offer on a more philosophical level. And indeed, quantum cognition has made interesting inroads into accurately modeling human behavior. very briefly, quantum probability models have been shown to more accurately model behavior that is seen as irrational. The example that I found most interesting was what is known as, in psychology, as context effects. That is, that the order in which you ask certain questions matters. For instance, if you ask people first whether Bill Clinton is honest and trustworthy, and then you ask the same thing about Al Gore, they will answer differently to the case in which you switch the order of the questions. Why this is interesting in the context of quantum theory is because of commutativity, which represents the order in which you measure physical observables, and is a fundamental principle in quantum theory. I'm not going to say any more about this as it's not my field of research, but I think it's very interesting. And for those machine learning experts out there, there was also a recent experiment that showed that quantum reinforcement learning models were better at modeling value-based decision-making on both a psychological and neural level. And with that, I'll conclude with a very open conclusion, because most of what I've discussed is purely theoretical and far more experimental evidence is needed. But I think it's a very exciting field that could potentially lead to all sorts of new therapies for healing the brain, as well as possibly contributing to our understanding of human behavior and ultimately perhaps consciousness. And then these are my image references. And that's the end of the talk.
Yeah, thank you very much, Betony, for an excellent talk. Thank you so much. I see that there are already some questions in the Q&A. Ilya, would you like to guide us through that?
Yeah, sure. So the question is, yes, thanks for the great talk. Would you know if there are any known connections between consciousness and light/photons?
Well, only in the fact that consciousness seems to be mediated by neurotransmitters in some sense, and that neurotransmitters are sensitive to light. So yes, I would say there is possibly. And as far as, I don't think there's a set definition of what exactly consciousness is, but if it is located in a biological system such as nerves firing or neurotransmitters, then yes, I do think consciousness is responsive to light.
Okay, thank you. The other question is, what do you believe consciousness is?
I specifically put at the beginning that I wasn't going to discuss consciousness because I don't have a fixed idea of what consciousness is. Read the Penrose and Hameroff, because they focus on consciousness. I wanted to present physiological, so how nerves fire, how neurotransmitters work, how general anesthetics work. So the level of my interest is more mechanical, but possibly if we can work out how these mechanisms work, we can apply it to the results of these mechanisms, like consciousness. So sorry, I don't have a fixed idea of what consciousness is. That's a very hectic question for a Monday afternoon.
Okay, thank you. And there is another interesting question: is there an ethical barrier in testing these theories?
I think in as much as there's any ethical barrier in testing medical theories. I think with the brain slices, they were slices. They weren't. Nobody killed a human to slice their brains. They were already dead. But yes, as much as any medical tests are done on living creatures, there's always an ethical boundary. But that's why I'm a theoretician, not an experimentalist.
Okay, so how does the brain stores and process information at the quantum level?
Well, I'm not. This would be. So the first two things I discussed with the electron transport. That's on the level of picosecond. So that's not going to be storing of information. That's just how nerves work. Also with vibration-assisted quantum tunneling with neurotransmitters. I mean, if neurotransmitters are implicated in information storage, which I'm sure they are, then vibration-assisted quantum tunneling would have some effect on information storage. But I think the most relevant thing that I discussed in terms of that question is this Fisher mechanism with where you get coherence times of an hour to days. But he doesn't elaborate on how to actually store the memories or the information. He just shows how you can get you can get nerves firing in correlated ways. He doesn't go further and explain how you actually store this information.
Okay, so due to quantum effects, can the human brain reacting to the stimuli be seen as non-deterministic?
Sorry?
Due to quantum effects, can the human brain reacting to the stimuli be seen as non-deterministic?
Well, I think when the interactions finally happen, so when you get, so say with the spin, you have the coherent spin states, say in the avian campus, but then when they actually end up recombining and going into a chemical product, that would be deterministic. It would depend, I mean, the ratio of spin states would depend on where they are in the magnetic field, but it would have a deterministic product, if I'm interpreting your question right.
I don't know, probably, but also one need also probably also to mention that the kind of like you can have a non-deterministic behavior completely classically due to just due to the statistics. So kind of one don't need to associate non-determinism to quantum only. Okay, so
Thanks,
the next question is, what does anesthetic substance do to neurons?
They don't really know. I mean, this is why this is an interesting field. This is why there's actually some scope for whether quantum effects have any role. So as I mentioned, in the framework of quantum effects, it's been shown that anesthetics change spin states in neurons. Classically, I think anesthetics change communication between brain areas. It seems to shut down the communication between brain areas, but they don't understand. I mean, it's an open question how general anesthetics actually work.
Okay, so how superposition of quantum states alters the mindset of the observer, if any?
Well, okay, so take these, take the Fisher hypothesis. So if you have, say you have, so you have the entanglement of or this correlated spin states, the covariance between the singlet and triplet states, and this is altered, say, by adding a lithium into these hypothetical Posner molecules, and you alter the entanglement. Then altering that entanglement alters the amount of binding that these molecules do, and that alters the amount of calcium, because when they bind, they melt, and that will alter the amount of calcium, and the calcium then has an effect on neurotransmitters, and neurotransmitters have an effect on nerves, and nerves have an effect on your mental state. So that's how you would get from these singlet triplet superpositions to an altered mental state, as far as I see it. So it would go through the
Okay, and there is kind of like a little bit open-ended question. You mentioned near the end that for now we have these theories. We need to have some experiments to back them up. What sort of experiments we are talking about here? So
there would be different. There would be different experiments for the different things. I think with the coherent electron transfer, they've been trying to. They've suggested that the same approach that they do to photosynthesis, electron transport chains in photosynthesis, they can do to these electron transport chains in time resolved. And I've totally forgotten what it's called, but there's lots of experimental results for photosynthesis, and they could just do the same for the tubulin examples, because all it is how energy moves through these pigments. So they could apply those experimental techniques to the neurobiological context. In the vibration-assisted quantum tunneling example, they would have to do more of these selective deuteration experiments. They've only done one, I think, for serotonin, one for histamine, which, yeah, they need to do a lot more to see, because I know some of the olfaction results were problematic because there were contaminants included, which changed the vibrational spectra, which changed the outcome. And then for Posner molecules, we were looking at doing an experiment maybe with seeing how magnetic fields could, because spin is responsive to magnetic fields, so seeing how magnetic fields could alter the spin states of these Posner molecules, and then alter the calcium production, and then alter nerve firing. So it would be different experiments for the different, the three different things I discussed.
Okay, great. And there is a comment from Camille. So in relation to light response of the brain, this reminds me of the elliptic attacks triggered by light. But there, the brain reaction might come from the signal from the eyes rather than directly from the light itself.
Thanks, Camille. Yes, I think there's actually people looking at how light's taken in back, because in the retina, it is the rhodopsins, which are also chromophores, and they're looking at how light taken in rhodopsins can have a knock-on effect in the brain. So yeah, I think epilepsy, light-affected epilepsy is also a very interesting open question for quantum effects in the brain. I think the most exciting thing about this field is that potentially you could get a whole new type of medical intervention out of it.
Okay, that's it.
Okay, Ilya, then thank you very much. If there are no more questions, it's time to thank you, Betony, for a fascinating talk that triggered lots of questions. And as you very bravely indicated several times, to most of those questions we don't have any answer, which means that there's still lots to be done and lots to be studied, but it's really very nice, and I hope that everyone really appreciated that you really try to distill out of vast literature of sometimes crazy speculations the few non-fake news, yeah, that might guide us in understanding if quantum biology or quantum effects play a role in neurobiology. Yeah. So Betony, thank you very much again for an excellent talk. Ilya, thank you for moderating so brilliantly the questions. And thank you very much to all the participants for your loyal participation and attendance of the NITheP Colloquia. And maybe some of you are also attending the NITheP mini school, so that will take place again tomorrow afternoon at 2. So Betony, thank you very much.
Thank you very much.
Have a good evening, and a good evening to everybody. And please keep staying safe and cautious, because we don't want a second wave of COVID to wipe us away. Yeah. So thank you very much, everyone. Have a good evening.
Thank you very much. Bye-bye.
Bye.